In this first module, we cover the anatomy of the basal ganglia and its functional circuitry because understanding biology is the first step to understanding function. We've all got basal ganglia, even these very ugly lamprey fish without a jaw that were here on Earth 500 million years ago before the sharks and amphibians and dinosaurs. There were the lamprey fish, which also have the basal ganglia. So this means that the basal ganglia was phylogenetically ancient. And what's more, the neurochemical markers, the cell types that make up the basal ganglia, its circuitry, are really very similar across different species. So it doesn't really matter if you're a lamprey, or shark, amphibian, dinosaur, bird, or mammal. You've got basal ganglia that's very similar. The basal ganglia has no direct input or output connections with the spinal cord. All of its influences are via the cerebral cortex or the brainstem. So this little diagram here, the blue areas here, this is the caudate nucleus, this thing here on top, and this region here is the putamen. And the caudate nucleus and putamen together constitute the striatum, which is the major input structure of the basal ganglia. If we take a coronal section of the brain, this is what you'll see. The blue sections are part of the basal ganglia. So here's again your caudate nucleus and your putamen, which form the striatum, the input structures. And then we have the globus pallidus externa, the external part of the globus pallidus. Globus pallidus simply stands for white globe. And then you have the globus pallidus interna, so the internal region of the globus pallidus. And then we have the subthalamic nucleus, which, as its name implies, sits below the thalamus. And we have the substantia nigra, which simply means black substance because the tissue looks black because of the high amount of neuromelanin in it. Areas of the cerebral cortex project in a topographic way onto the striatum. And there are different functional circuits that are segregated from each other. And output in each functional pathway is projected in a somatotopic way to different parts of the thalamus. And from the thalamus, it then projects to the cortical areas from which the circuit started. So in this way, it partially closes the system of cortico-subcortical loops. The basal ganglia actually receives inputs from almost all parts of the cerebral cortex, but for the motor loop, the basal ganglia receives inputs from the motor cortex, the supplementary motor area, and the premotor cortex, as well as other sensory regions of the brain. So here is those projections to the basal ganglia. These are the basal ganglia nuclei. And the basal ganglia outputs back to these cortical regions from which this loop originated. It outputs back to these regions always via the thalamus. So this is the motor loop that I've described in the previous slide. So what are the effects of the basal ganglia outputs to the thalamus? Well, it's first useful to know what the thalamus does to cortex. The thalamus is like an overexcitable puppy. It always wants to excite motor cortex to make movement. The basal ganglia, or more specifically the globus pallidus interna, acts like a leash for this overexcitable thalamus. This is because the globus pallidus interna's output to the thalamus is inhibitory. It wants to silence the thalamic output. Within the basal ganglia, there are two pathways. The direct pathway, which goes directly from the putamen to the globus pallidus interna, GPI, and the indirect pathway, which goes from the globus pallidus externa to the subthalamic nucleus, and then finally to the globus pallidus interna. And this indirect pathway inhibits movement, while the direct pathway facilitates movement. And these two pathways together modulate the inhibitory output of the basal ganglia to the thalamus. So the basal ganglia works by a balance between these two facilitatory and inhibitory pathways. So remembering that the thalamus has an excitatory effect on the cortex, and the globus pallidus interna, or the leash, inhibits the overexcitable puppy, that is the thalamus, we can now look at the effect of the direct route. So when cortex sends an excitatory input to the putamen, it has the effect of inhibiting the globus pallidus interna. So effectively, the leash holder, the globus pallidus interna, is now tired, and this reduces the inhibitory effect of the globus pallidus interna to the thalamus. And because the thalamus is now less inhibited, it can go ahead and activate the cerebral cortex. The indirect route has an opposite net effect. Activating the indirect route inhibits movement. So first, the cortex sends an excitatory output to the putamen, which then inhibits the globus pallidus externa, which then inhibits the subthalamic nuclei, and that has the net effect of disinhibiting the globus pallidus interna. So the globus pallidus interna goes, cool, I have lots of energy to hold on to the leash tightly, and this then increases the normal inhibitory tone or the inhibitory effect of the globus pallidus interna to the thalamus, and this results in less excitation of the thalamus to the motor cortex. Let's look at the effect of dopamine in the basal ganglia. So dopamine is released by the substantia nigra, and it binds to two different types of dopamine receptors in the putamen. Dopamine has an excitatory effect to striatal neurons that project to the direct route. So the direct route facilitates movement and is actually facilitated or excited by the dopaminergic projection. In contrast, dopamine is inhibitory to the striatal neurons that project to the inhibitory indirect route. So because the indirect route is inhibited by the dopaminergic projection and the direct route is excited by the dopaminergic projection, the net effect of dopamine is to facilitate movement. So this is a summary slide, hopefully to help you consolidate your knowledge from this module. So the basal ganglia receives input from the cortex via the striatum, and it sends inhibitory output from the globus pallidus interna to the thalamus, which has an excitatory projection to the cortex. Thalamus is like an overexcitable puppy that always wants to excite cortex. With that knowledge of the relationship of the thalamus to cortex, that excitatory relationship of thalamus to cortex, and that inhibitory relationship of the globus pallidus interna to thalamus, you can then build your understanding of the two primary pathways, the direct pathway and the indirect pathway. So in the direct pathway, cortex first excites the putamen, and then the putamen then inhibits the globus pallidus interna. So effectively, the leash holder, the globus pallidus interna, is now inhibited, is now tired, so it can't inhibit the thalamus as strongly, and then the thalamus is free to go and excite the cortex. So that has the net effect of facilitating movement. For the indirect pathway, an excitatory output is sent from the cortex to the putamen, which inhibits the globus pallidus externa. And the globus pallidus externa inhibits the subthalamic nucleus, which then goes and excites the globus pallidus interna. So the leash holder has lots of energy to hold on to the puppy tightly. Um, and then as a result, the poor little puppy is inhibited, the thalamus is inhibited, and consequently is less able to excite cortex, and this results in movement inhibition. So dopamine facilitates movement by facilitating the direct pathway and by inhibiting the inhibitory indirect pathway. So the net effect of dopamine is to excite, to facilitate movement.